Earlier you learned about one type of cell division, which was mitosis, which is used for asexual reproduction, and in animals, as we learned, to repair tissue, embryonic development, and things like that. Now, humans have 23 pairs of chromosomes, or a total of 46 chromosomes. When a human cell undergoes mitosis for tissue repair, you start with 46 chromosomes and you end with 46 chromosomes. What we're going to learn about now is a type of cell division called meiosis, and the goal is to go from those 46 chromosomes to 23. So these are both called diploid numbers of chromosomes. Di meaning 2, because you have 2 of each of the 23. And when you get down to 23, that's called haploid. And what that will allow is meiosis is what produces gametes that can be used for sexual reproduction. And there's a lot more variation with sexual reproduction, but we'll get into that in a little bit. So as we look at this, here is our homologous pair of chromosomes, and we know that during interphase they double, and so we have sister chromatids here that are matching for that, and here's 2 sister chromatids. And in fact, because this is a pair of homologous chromosomes and we have sister chromatids, we call that a tetrad because there are 4 sister chromatids. And as mentioned, we have 2 different chromosomes here and a homologous pair. So 2 chromosomes, or another way of putting that is a homologous pair. Now note, right here, we also have 2 chromosomes and a homologous pair, only there's 2 sister chromatids here, where there's 1, 2, 3, 4 right there. In meiosis, the first thing, we have many of the same phases that you have in mitosis. When they line up in the center, just like in mitosis, that would be called metaphase. And when they line up in the center here, these chromosomes that are homologous pairs hang out together, that tetrad hangs out together, and every once in a while when they cross over, because they're so close, a bit from one chromosome will go to the other, and that's called crossing over. So the first thing that happens, and this is called meiosis 1, is that homologous pairs separate. Now we're down to 1 chromosome in each cell, 2 sister chromatids in each cell here, and we do not have a homologous pair anymore. And if you remember, in mitosis, you always have homologous pairs. It's the sister chromatids that separate. Well, in meiosis 2, which is the second division of meiosis, that's when these sister chromatids split apart. So now at the end here, what started as 2 chromosomes, a homologous pair, we refer to that once again as diploid. We now have 1 chromosome, no homologous pairs, and that's referred to as haploid. And so what you really have in many ways is an incomplete cell. And the only way that cells can be completed is if it combines with another cell. So these cells are called gametes. And that process down the road, when they meet up with another cell and become whole, is called fertilization. And that is part of sexual reproduction. And also, if you notice here, none of these gametes looks exactly like the parent cell that you started with over here. They all look just a little bit different. And that's how meiosis can lead to a lot of variation. So in summary, in meiosis, you start with diploid cells that have homologous pairs. As you go through interphase, that's our G1, and then it's the S phase. We know that when things duplicate, you still have diploid cells. you still have homologous pairs, and that is when you end up with four chromatids, sister chromatids, which are known as a tetrad. Then we have meiosis 1, where the homologous pairs line up. There's some crossing over that occurs. That's where genetic information can be swapped between sister chromatids. And they split apart. And as they split, you are now haploid. And you do not have homologous pairs. Then in meiosis 2, sister chromatids split. So there's a quick summary of what happens in meiosis for you.